on the future of what soil health could be if it continues to be a thing. And this is where everyone starts. This is the introduction to soil health. So just doing a basic definition of it, soil health is the capacity of soil to perform ecosystem services that sustain productivity in its soil. And basically those soil ecosystem services come from the physical, chemical, and biological domains of the soil. For example, they're regulated vital processes, and these are some examples here. For example, water filtering. The soil acts as a filter for water, filters out those nutrients and provide them to crops. Then you have soil aggregation, where if you have carbon, for example, carbon above the soil that decomposes and aggregates to form aggregates in the soil. And then you have nutrient cycling that acts on the surfaces of the soil particles. And then, of course, everyone wants to know about carbon storage in the soil and how soil health affects that. Carbon storage comes from all the biological life in the soil that decomposes and then leaves behind carbon residue. So we have these, and there are many more. We have these as some of the main processes that happen in the soil that are related to soil health. And the way people usually recommend sustaining these processes is, one, through maintaining roots in the soil, because those roots stimulate the fauna in the soil, and the flora stimulate the fauna, and then they act on each other and stimulate these processes. So maintaining roots throughout the year is important, because without those roots, a lot of these processes won't continue going. Then you have providing cover. So for example, if you have a cash crop that's harvested in the summer or whatever, and you leave the soil bare for the rest of the year, well, that's contributing to a detriment in terms of these soil processes. So providing cover throughout the year is important, and people usually do that by having a cover crop in their soil. And then diversifying plants, that's another way to improve upon the soil health processes, because if you have diverse plants, then you can have diverse functions in the soil. The main diversity being the corn-soybean rotation, where one year you have your grain and your grass crop, and then the next year you have your grain and your legume, which has two different functions in the soil. For example, the legume provides nitrogen, and then the grass crop has a C4 productivity for carbon. So there's that dynamic there where you're contributing to diverse function in the soil and therefore diverse microbiota. And then one of the most important things is reducing tillage or reducing disturbance of that system. Because if you have your roots throughout the year using your cover and diverse plants, if you're constantly disturbing that system, it probably won't mean as much because it's not long lasting. So it's important to reduce tillage or reduce disturbance in that system so that you can sustain soil productivity throughout the year and also many years. Now, these are the four main recommendations for sustaining soil health and productivity. And one of the challenges of soil health is that it's not a directly measurable context or a concept or property of the soil, because you can't just probe the soil and say, this is soil health. You have to integrate many different factors of the soil in order to learn what the soil's conditions are or what the soil's health is. So I just have some examples listed here where there's the biological soil health or soil health indicators, which is what we call them, and those are all based on the vitality of the soil or life in the soil. For example, if you measure organic matter, you know that organic matter in the soil is contributed from biological life. So that gives you an idea of how active or how much biological life is in the soil, because more organic matter typically means there's more biological life there. And also CO2 respiration. That's another factor where the microbes in the soil, they consume carbon and then they respire, and you can measure that respiration as an aspect of how active life is in that soil. And then you have your biomass. Biomass can be contributed through either animals or plants. So the more biomass you have in the soil, the more life you typically have in the soil. And then there are bioexudates, so that animal and plant life and microbe life all exude chemicals or biochemicals into the soil that, based on how they live, tell you what's going on in that soil. So those are all the biological aspects of soil health indicators. Then we have the physical soil health indicators, which are typically based on how malleable or ductile or resilient the soil is. So for example, if you were to measure soil aggregation, that gives you an idea of how carbon has aggregated that soil and how well both the compaction and hydrology properties of that soil interact with the environment. Well, if you have poorly aggregated soil, you could have really massive soil that doesn't have good infiltration or porosity. or it doesn't have good, or which also means it doesn't have good hydrology, so water isn't moving through that soil. So all these physical properties interact with each other in some way. And then, of course, we have the chemical aspect of the soil, which is based on fertility. Most people are familiar with that aspect of the soil because it's typically what people do when they're trying to grow a crop on the soil. The fertility aspect tends to be the most important, and that's usually the mineral nutrients or pH or other conductivity aspects of the soil. So these are just a few examples, and I'll go through some detail of one example later. But I just want to tell you that there are many people looking at different aspects of the soil. There are many more aspects of soil indicators than those I just listed, and some institutions that are working on that are the NRCS, that's the Natural Resource Conservation Service in the U.S. They're kind of the dominant in terms of looking at soil conservation and soil practices. And then Cornell, they have their Cornell Comprehensive Assessment of Soil Health. That's sort of the leading soil health assessment in the United States right now, so I wanted to discuss that. And then there's the Soil Health Institute, which is an institution that looks at different research and other aspects of soil health, and they recommend stuff for the future of soil health assessment in the U.S. or the world in general. So these are kind of the major institutions that are influencing soil health right now, and I just wanted to point them out. And also I wanted to point out that soil health is not just about agronomic productivity. There's also a focus on environmental, ecological productivity. So my presentation today will focus on agronomic productivity, but just be aware that there are some other aspects of soil health that are unrelated to agronomic practices. But today, my research focus, or my presentation focuses on the Comprehensive Assessment of Soil Health from Cornell, because that's the leading soil health test right now, and that's what I use for my research. It's one of the tests I use for my research. And on this test, they have 12 soil health indicators. They have 4 physical, 4 biological, and 4 chemical indicators, and I'll go into more detail about what those are later. But just to quickly list them off, there's available water capacity, surface hardness, subsurface hardness, and aggregate stability as the physical indicators. The biological indicators are organic matter, CO2 respiration, protein content, and permanganate oxidizable carbon. And then the chemical indicators are phosphorus, potassium, minor nutrients, and pH. Now what Cornell does is take each of these indicators, and they have a collection of soil samples that they've measured each of these indicators on, and then they create a statistical distribution and an index to evaluate soil health. So their evaluation is based on the soils that they already have in their lab. And what they do is take the results from those and put them on a 0 to 100 index scale so that they can evaluate soil health, if you look at this chart here, from very low to very high soil health. They do this for each of their descriptors, and they also do an overall soil health score by averaging the scores from all 12 into one single score. And that's because, as I said before, there's no single soil health measurement. It's not a single measurement that you can do. So this is Cornell's solution to trying to evaluate soil health through integrating several properties into one score or evaluation. And just a quick overview of how they do this, or measure each of these indicators. These are the physical indicators here, the available water capacity. I should step back and mention that what you do is collect a soil sample in a field and then send that into Cornell's lab for measurement. So once you collect your soil sample, you package it up, send it to Cornell's lab in Ithaca, New York, and then they'll run all these measurements on it for you. That costs, I think the current cost is still around $110 to do all these assessments. So it is costly compared to a typical or standard soil fertility test, but the assumption is that you'll get more information out of this that'll be useful for crop management. So we'll have some opinions on that a bit later. They measure the physical indicators, one, available water capacity using a pressure chamber. So once you send the soil sample, they'll break it up and then apply pressure to it to see how well that soil holds on to water. And then the penetrometer is what's used to measure surface hardness and subsurface hardness. These are two measurements that you do, or the submitter does in the field before sending that soil sample. So you'll measure the penetration resistance in the field and then collect a soil sample and send it to Cornell. And then aggregate stability is measured using a wet sieve. So you take a soil sample, you put it in a sieve, and then dip the sieve in water and see how well that soil holds together under, being saturated with water. You typically very clay, lumpy soils degrade away, and then when you have the loamy, aggregated soils, those typically can hold together if they have lots of carbon in them. So you can see a difference there. And then the biological indicators, this is where things typically get more complex. The first one is organic matter, where they measure it using loss on ignition, which is the technique of taking a soil sample and then using a very high temperature to burn off the organic matter. You can get an evaluation of organic matter that way. And then there's a CO2 respiration, which is a 96-hour incubation. So they dry down a soil sample, re-wet it, and then wait 96 hours to see how much CO2 has evolved off that soil sample. That gives you an idea of the biological life in the soil or biological potential in the soil. And then there's protein content, which is a measure of amino in the soil or amino acids in the soil that gives you an idea of how much protein is there. And then there's the permanganate oxidizable carbon, which is a measure of what we interpret as the readily available carbon food source in the soil. So just different measurements of biological activity and life in the soil here. And then the chemical indicators are measured using your standard extraction or pH assessment. For Cornell, they mostly use the modified Morgan because that's popular in the Northeast. However, I've listed all these indicators, I've told you all these things about soil health, but another challenge that we have, the first challenge I mentioned is that soil health is not directly measurable. The second challenge is how is it interpreted in different environments? Because, for example, if I were to measure soil health in a desert environment or a savanna environment or a grassland out in the Midwest if I was in the U.S., or a forest, a temperate forest or a tropical forest, how do you interpret soil health in these different environments? Would you have the same standard across the board, or would you change your standard based on where you are? And for example, us in the United States, here, we're in Maryland, which has a dominance of ultisols. But if you were to go across the United States, you start running into grasslands and the mollisols here in the Midwest, and then you start hitting some iridosols out in Nevada and California. So if you were to measure soil health, you would probably have different interpretations of soil health as you grow across the land, because each of these lands grows different types of crops, has different type of ecology. So how do we assess soil health if we know that ecology and climate changes as you encounter different environments? And that's what led me to my research in North Carolina. So the Cornell soil health test was developed mostly using Northeast soils, but us people in the South, or when I was in the Southeast, we wanted to see how does this work for our soils. And this is kind of a physiographic map of North Carolina that I'll go into more detail later. But as I said before, I focused on the agronomic aspects of soil health management in North Carolina. So our objective for that research was determining if soil health indicators have the ability or can differentiate agronomic management in North Carolina. And this is just a quick diagram of how our research was structured. First, we identified locations in North Carolina where we wanted to collect the soil samples. And then we identified the soil health test that we wanted to use. For our research, we used the Cornell test, we used the Haney test, and we also used the standard North Carolina fertility test, which is the Mehlich 3 extract. And then we collected and submitted our soils for sampling using a 6-inch depth. So all of our soils are collected at 6 inches, so we're only looking at activity within that 6-inch depth. And then we analyzed the soils and compared our recommendations from the soil health tests. This is a map of where we collected samples from in North Carolina, just looking at the physiography of North Carolina. We had a mountain location, which is here in the rocky area of North Carolina, where we have typically rocky soils. And then the Piedmont area, where you have some clay soils. And then the coastal plain area, where the soils get a little bit loamier and sandier as you go out to the ocean. And there are some differences at the 3 locations. I'll briefly go over those differences. The mountain locations started in 1994, the agronomic trial at least, and that was about 20 years before we collected those soil samples. So there's a 20-year management history before we collected our soil samples there. There's a soil series, silt loam. Here there were very vegetable crops and sweet corn, and we had treatments that ranged in intensity from no-till to disc tillage, and we had organic management and conventional chemical management there, and we also had a cover crop at this location. And in our Piedmont location, that trial had existed about 30 years before we collected our soil samples. So it's a different soil series than the mountain location. Here is a typical corn and soybean rotation. And we had a difference in tillage intensity ranging from no-till all the way to moldboard plowing. So, very wide range in terms of how much the soil had been disturbed over the years in our treatments there. But there was no cover crop there in the Piedmont location. And then in Goldsboro, which is the coastal plain location, that trial had existed about 15 years before we collected our soil samples. It's a different soil series than the other two locations. There are vegetables, corn and soybean grown there. We have differences in tillage intensity. And here we had a wide range in cover crops from the legume cover crops to the grassy cover crops. So you can see here that our trials are different, and we're hoping to pick up different aspects of soil health from these three trials. And now that I've kind of described everything to you, I'll just go over the results. So just be cautioned that it's a lot of information coming to you at once, but I'll slowly break down everything and go through the data. So this is how I'll organize all the data for you. At the top is an image of what location it is, and followed by the treatments at the location. So this is an example of the coastal plain location where we have the no-till tillage, disc tillage with conventional chemical management, and then disc tillage with organic management. And then at the bottom is going to be a table with the treatments listed in the leftmost column, followed by the 4 physical indicators: available water capacity, surface hardness, subsurface hardness, and aggregate stability. Then the biological indicators: organic matter, soil proteins, CO2 restoration, and active carbon. And then the chemical indicators: pH, phosphorus, potassium, and minor nutrients. And then the far right column is our overall soil health score from the Cornell assessment. So remember, this is the score where they average all of these 12 scores together and give us an overall score for soil health based on the soil sample that we submitted. And within the table, I have letters that indicate when there's a statistical difference between the treatments at the location. So for example, here at the coastal plain, when we look across all the table columns, we see that there's no statistical difference except for where active carbon was greater in no-till compared to the conventional tillage treatment with no conventional chemical management, and the conventional tillage treatment with organic management. So it's about twice as much active carbon, which means twice as much readily available carbon food source at that location. And that's obvious because no-till typically leave the carbon on the soil, and then there's more food for the microbes. So that makes sense. However, when we look at the scores, or at least the ratings from Cornell, I have the color indicators here where, remember, red is the very low score, and then green is the very high score, and you have intermediate color in between there. So at our location here, for the physical indicators, we have surface hardness, subsurface hardness, and aggregate stability all scoring low or very low for all of our treatments, including the no-till. Now remember, no-till is considered to be important because we want to reduce the physical stress on the soil, but when we look at the actual rating that we got from Cornell, we were still scoring very low soil health based on their assessment. And that's an issue because we'd been doing no-till at this location for 15 years before collecting these soil samples. So it brings up the question of how long do we need to be in no-till at this location before we start getting positive scores from the Cornell assessment. So that's bringing up an issue there. When we look at our biological indicators, although we had a difference between the no-till and the organic and conventional plow management, when you look at all the ratings from Cornell, all of them were very low according to their assessment. So again, even though we have comparisons between no-till and disc tillage, we're not getting that differentiation based on what we recommended for soil health management on the previous slides. And then our chemical indicators, there's plenty of phosphorus, plenty of potassium, plenty of minor nutrients. Although our pH score was very low, I think that may be because in the southeast the soils have typically different type of pH compared to what you'd expect in the northeast based on the modified Morgan. So there's some dynamic there where our pH score is very low, but I don't think it's very meaningful because it's a different assessment than usual. When you look at our nutrients, there's plenty of nutrient cycling going on there where we have plenty of nutrients compared to what we'd expect from these systems. And then our overall scores were low or very low based on this assessment. So we're comparing no-till, organic management with tillage, and conventional management with tillage, or no-till with conventional chemical management. And we're not getting high scores for any of these systems, which is questionable considering the no-till that we have there. And then I'll move on to the next location, our Piedmont location, which had the intensive tillage going from no-till all the way to moldboard plowing. The table here is laid out the same way where we have our physical, biological, and chemical, and then overall score at the end. these are the letter indicates where you have differences. And as you see again, there's no statistical difference in our physical and chemical indicators, but when we look at our biological indicators, we have more soil protein in our no-till system than we do in our double-tilled systems. So that's where we have a chisel and disc plow going through every year, or a mower and disc plow going through every year. So we saw that intensive tillage had degraded soil protein, and also we saw that there was more CO2 respiration where we had a disc till or a chisel plow compared to the others. And that also makes sense because when you have no-till, it's less likely that you'll have lots of CO2 respiration because you're not disturbing the soil compared to where you're tilling. You disturb the soil and you encourage respiration. But again, when I look at the actual ratings from Cornell based on how they're interpreting these scores, our physical indicators all scored low or very low except for available water capacity. And then our biological indicators all scored low. And our chemical indicators all were sufficient. So we had enough phosphorus, potassium, and minor nutrients in that system. And then you look at our overall soil health scores, and we have low or very low. So again, we're seeing the same trend where none of our biological indicators are high, none of our physical indicators are high, but our chemical indicators are sufficient, which means in these systems we're doing pretty well on the actual nutrients that produce a crop, but we're not doing well on these other aspects of what we're considering to contribute to soil sustainability in terms of physical and biological soil properties. And then when we look at our other location in the mountains, this is where we had the most distinct treatments. We had no-till with organic management, which is probably considered the apex of what you could do for soil health because you have the organic amendments coming in, you have no-till, which doesn't disturb the system, you have a cover crop, so we had everything there. And then we have the no-till with conventional chemical management, with conventional pesticides and stuff, so it's not the best, but it's pretty good. And then we also had the disc plow with chemical management, disc plow with organic management. And at this location, we had a treatment that was specifically designed so that we did not apply fertilizer in any years. So this is just, we disked the soil every year for 20 years and then measured it at the end to see what would happen. Again, our treatments are on the leftmost column, followed by physical, biological, and chemical indicators with overall at the end. Here you'll see again we have a difference in active carbon where there was more active carbon in the no-till organic system compared to our tilled systems, which makes sense because if you have those organic amendments every year, you're definitely contributing a lot of carbon to the soil, and that causes lots of microbes to be active and respire, and that you have that readily available food source there. But again, when we look at our actual ratings or interpretations from Cornell, our physical indicators were low or very low, our biological indicators were low or very low, and our chemical indicators were high, except for pH. And it's important to notice here that for our treatment where we did not add fertilizers, we still had enough nutrients cycling in that system to have sufficient phosphorus and potassium after 20 years of just tilling that soil and not adding fertilizer. But when we look at our overall scores this time, I do want you to notice one difference. Here is the no-till organic system where we finally have a score that's medium soil health. So even with no-till organic, based on Cornell's interpretation, the best we got after 20 years of no-till management with organic amendments every year, I think it was poultry litter every year, we still had medium soil health. So that starts to bring up questions of what do we need to do here to get high soil health? What interpretation would Cornell have to have a high soil health management system, or at least agronomic management system? No, that's the only treatment, no-till organic. When we look back at all the data, the only indicators with differentiation are CO2 respiration, protein content, and active carbon. Now, for those of you who aren't familiar with those measurements, it's not something a standard soil lab would do. So you wouldn't typically see these measurements just hanging around at a soil lab for them to process them and see what's going on. But you also notice that all of these are biological indicators. So each of the indicators that actually had some statistical differentiation were biological indicators. And then when we look at all the indicators that did not differentiate, well, we had 12, only 3 differentiated. So we only have 25% of the indicators actually meaning something when we're looking at differences in systems approach to agronomic management, which is an issue. So when we look at the reduced tillage, providing cover, maintaining roots, and diversifying plants, these are all the recommended management practices for soil health management. We had reduced tillage at 3 locations. We had providing cover at 2 locations. We had maintaining roots at 2 locations. That's the providing cover and maintaining roots is basically having a cover crop during the winter after you harvest your cash crop in the summer. And then we had diverse plants in 2 locations where we had vegetable crops on top of the typical corn soybean rotation. But even though we have these practices at our locations, after 15, 20, and 30 years, our soil health score is still low or very low based on this Cornell assessment. Now, I'm saying this not to say that Cornell is wrong. I'm just saying that the interpretation is not what you would expect if you were to do these practices for many years or over several years. So that starts to bring up questions of what are we actually getting from this data? What does it mean when we have low soil health, even though we've done no-till, we've done organic management, we've done all these things that people are recommending for soil health, but we're not getting the results that we want or expect. And that's where I'm going to get to the part that most people care about, or at least most farmers care about, which is long-term crop yields. Because as I just said, if we don't know what we're getting from these soil health analyses or soil health indicator analyses, what does that mean for the farmers? And that's where we start connecting it to our actual management goals, one of them being crop yield. Now this is, again, a lot of data to throw at you at once, so I'm going to break it all down for you. These are the 3 locations that I've been discussing for the past few slides. We have the mountain location, the Piedmont location, and the coastal plain location. And we have the yields here at the mountain location where I have treatment intensity or tillage intensity going from top to bottom. So no-till is going to be at the top, and then the most intense tillage is going to be at the bottom. At the mountain location, we had sweet corn, and I have it listed here in bushels per acre, but that's a little misleading because it's not green corn, so don't interpret it the same way as you would green corn. Here, you'll notice that the no-till treatment and the tillage treatment yielded more based on statistical groupings than the organic treatments. But that's a little misleading at the mountain location, to be fair, because we did not manage these organic plots as one would typically manage in terms of always being there and picking every weed and being laborious about the whole process. So this, I don't think, is a soil effect, even though we had more yields from the non-organic plots. I think it's more of a management effect than it is a soil effect. So I wouldn't consider this a difference in soil health yields. But at our Piedmont location, which is where we had differences in intensity of tillage, I do think it is a management effect, because here we had grain corn yields and soybean yields. And you'll notice that in the no-till treatment, the grain corn yield is almost twice as great as what we got from the moldboard plow treatment, probably like 75% greater. And I think that's important there because it shows you that long-term, the no-till had performed better than our typical tillage or conventional tillage that the farmers were doing, for the 30 years. Remember, this experiment started in 1984, which is when farmers were still pretty much always tilling their soils. No-till became a little bit popular after that. And I can show the farmers after this experiment that long-term no-till typically yields more than their typical tillage practices. And that was pretty good. Now, it was a little different for the soybean, and I think that's an effect of the physiology of soybean. But again, we had greater yields. We had twice as much yield from no-till soybean than we did from moldboard plow soybean. So you can see there that there is an effect, even though it's not always statistical difference. And then at the coastal plain location, we didn't have the significant effects that we did at the other locations, but I think that's because this coastal plain location is typically overburdened with lots of drought in some years, and then in other years it's flooding because of the coastal flooding. So it changes up every year. So this location is a little bit complicated. But I want to break down the data even more, because this is where I'm going to connect soil health to crop yields. Now here on the chart, I have the overall soil health score by plot based on Cornell's assessment, and on the y-axis I have the grain corn yield for the Piedmont location, and over here I have the sweet corn yield for the mountain location. The main thing I want you to get from this is that if you look at the range of the overall soil health scores from Cornell, they go from about 30 to 60 on their 0 to 100 scale. But our grain corn yield goes pretty wide in terms of distribution. So the message I got from this chart is that the sensitivity of the Cornell soil health assessment is not picking up our differences in potential crop productivity. Because if you see here, our crop productivity has a wide range, but our Cornell assessment scores have a pretty short range. So clearly, there's some sensitivity there that's not being picked up by Cornell's assessment. And we had the same trend at the other location where we were growing sweet corn. So for sweet corn and green corn, we weren't seeing those trends. And then when I break the data down even more for individual indicators that we have on the assessments, organic matter, that one did, well, let me back up a little bit. So the green dots on these charts indicate where there's conservation tillage. The yellow triangles indicate where there's moderate tillage, so we just did a single pass of a disc plow. And the X is marked where there's intensive tillage, where we went through with the moldboard plow and then followed up with the disc plow. And what you'll see here is that there are some trends when we look at the individual indicators. For example, organic matter doesn't have a strong trend, but if you look at protein content in the soil, as protein content increases, you typically have an increase in corn yield. And when you look here, you see that a lot of the conservation tillage is where there's more protein content. So there is a signal there, it's just not a very strong signal. And we see the same trend with permanganate oxidizable carbon, which is the readily available carbon in the soil. Again, lots of the conservation tillage has more of that carbon, but when we look at the actual trend, it's not very strong. And then CO2 respiration, where we're looking at the activity of the microbes in the soil, lots of the conservation tillage has the most CO2 respiration, but again, when you look at the overall trend, it's not very strong. So I think what this is showing is that, yes, these biological indicators may have some potential to tell us more about these soils beyond just typical mineral fertility, but the signal is very messy. And that's where I'll summarize the results by just saying that the indicators were not sensitive enough or precise enough to show these differences in soil health. And we also did this using the Haney soil health test, and we got the opposite result. The Haney soil health test actually said all of our soils were healthy, compared to the Cornell test, which said pretty much all of our soils were not healthy. So just comparing between these two tests that are popular for assessing soil health, we're getting different results. And then when we did our typical North Carolina fertility test, this is what farmers respond to the most. They just needed to know if they needed more phosphorus or potassium or sulfur. And that typically resulted well for the farmers, or at least the researchers who were doing their research. Yes, go ahead.
So what region is the Haney soil test generally looking at?
I don't think Haney has a specific region. It was one of the initiatives by NRCS probably 7 or 8 years ago, and the Cornell test has kind of taken over since then. So there's no specific region. The Cornell test was developed in the Northeast, though. So basically what I learned from all this, I'll go through this quickly. The soil health indicators do not consistently vary in response. The biological soil properties tend to have a more extreme response, but still variable. And then I don't think soil health, as we currently interpret it, has a significant correlation to crop yields, or at least agronomic productivity. And based on these results, we recommend calibrating measurements so that we can adjust for sensitivity in the measurements, and then scale the relevance of our soil health assessments so that we can know more about what we're getting from these data, and then integrate crop yields when that's an important part of what you're measuring for soil health. But I want to break everything down here and go with the overview of what I learned from this research as I was doing it. So this image here is of a no-till field in one of my studies next to a disc-tilled field. So this field here was disc every year. This field has been no-till for 30 years. And you can look at this field and see a clear difference between these two areas. This one, it's a little weedy, but overall, the soil is pretty healthy. And this one, it's pretty clear soil. But what I want you to know is that when I walk into this field after a long drought or a heavy rainfall, I can walk on this side of the field pretty easily. I walk through that one and it's very comfortable to collect soil in that one. But after a long drought or a heavy rainfall, this soil here is almost difficult to collect. It's not even worth trying to get in there because you have mucky boots if it rained, or if it's really dry, it's all clumpy. So that kind of gave me the sense of what the problem is here, because when I walk in that field, I can feel a difference between the soil properties, but when we collect the soil and measure it in a lab, we were not able to quantify that difference. And I think that's where we're having trouble here, because as many of you may know, NRCS sometimes focuses on the, not the scientific aspects, but the promotional aspects of their practices. And that's what we're running into as an issue here. I definitely feel that difference, I see that difference, but I don't know how to measure that difference yet. And that's the issue I have. We don't know how to quantify how to make progress in soil health. And so based on what I learned from this research, I just have a few recommendations for the future. So we have the qualitative versus quantitative issue where I just mentioned I can feel the difference in soil health, but I have trouble measuring it. And then we have the minimum data set issue where we don't know, for example, Cornell, they have 12 soil health indicators, but I just showed you that only 3 of them actually made a difference in our, or 3 of them only showed differentiation in our plot. So what's the number of indicators that we should actually be using here? Because more is not necessarily better in terms of what we're trying to interpret from these soils. And then we have the regional importance. How do we differentiate which indicators or which soil properties are more important in different regions, and how do they affect our interpretations? And I just wanted to show this last chart really quick before I stop. And, well, actually, I may be able to go into the NRCS stuff one. But just to show you an overall view of soil health here, this is a chart by Norris and Congreve published in 2018. They did a review of vegetable crop production across many fields, and they have the relative response here. This is the soil health indicator relative response. This line here indicates the one-to-one line if you had the same response from plots that were not managed as soil health plots and plots that were. And the soil health properties that are practices that they looked at are organic amendment, cover crop, and reduced tillage. Now, these first 4 properties here, bulk density, porosity, soil moisture, and aggregate stability, they all respond well to adding organic amendments. These next properties here are the biochemical properties like total carbon, total nitrogen, and those all respond well to organic amendments, cover crop, and reduced tillage. So you can see here that there is an effect of these practices, the reduced tillage, the organic amendments, and other stuff, adding a cover crop. There is an effect here, because if there wasn't an effect, you would see everything either on this one-to-one line or less. And this is where things stand out. This is the biological properties. So again, when I showed you my data, I showed you that a lot of the biological properties responded much greater than the other physical and chemical properties. And that's what they show here. They show that microbial biomass, carbon and nitrogen, significant effect there, off the charts. The respiration from microbes, significant effect there. For the most part, there's one here that didn't have an effect. And then other things like enzyme activity. So all of these are greater than that one-to-one line. So that means when you implement these practices, relatively, they respond better. The soil responds better. But one story that I always run into is the cover crop issue. This chart here shows that if you add a cover crop, then the relative response in weed biomass and vegetable crop yield is worse than if you didn't have a cover crop. But one thing that I want to point out is that when we're talking about soil health, we're not always saying that your soil will be, or your soil will yield more. That's not what soil productivity is. When I'm discussing soil productivity, I'm talking about long-term productivity. So yes, this cover crop may have increased weed biomass. It may have increased or decreased vegetable crop yield. But as I showed you, that long-term no-till research that I had, or long-term no-till plot that I have in my other study, in the long term, those yields were much better than the other stuff. So if you have no-till and cover cropping, that generally in long-term studies is better. However, it doesn't mean it's going to yield more. It just means it's more sustainable. And if a farmer is focused on sustainability, then they're looking at these things. If they're just focused on year-to-year yield, they're going to look at not having cover crops, not having no-till. But again, when I showed you in that other slide, when I walk into my plots that are no-tilled and cover cropped, then I have much better time getting into those plots on a year-to-year basis than I do on that field that was distilled every year. And I'll just leave you with this one, because this is where NRCS has responded to those type of comments. They've come up with criteria for evaluating soil health indicators. And the first one they have as soil health indicator effectiveness. How effective is that indicator relative to not having these management practices? And they're focused on management sensitivity, short term, well, 1 to 3 years, and it has to be interpretable. So if I were to tell a farmer that they have this much active carbon in their soil, the farmer should be able to understand what that means and what they should do to improve it in the future. So that's what this means. And then we also have the effect of production readiness. How easy it is to measure that indicator, how much cost effective it is. Those are 2 things that are important for evaluating these. Then we have repeatability, how easy it is to repeat those results. So if I measure it one time and get a result and measure it another time and get a completely different result, then that's not repeatable because it's giving me different results. And then this is probably the most important for the CCAs here, interpretable for agricultural management decisions. If we're looking at soil health, if I tell you to do no-till, if I tell you to do all the cover cropping and diversify your crop rotations, how does giving you this data in terms of active carbon and CO2 respiration, how does giving you that information help you make better agricultural decisions? And I think that's an important goal that NRCS is now focused on, because if we keep telling farmers to do different things and they don't see the results, they may not understand what's going on, and that's an issue. So this whole soil health thing is kind of confounded by our interpretations of environmental sustainability compared to agricultural sustainability and production. So that's what I want people to start focusing on in the future, and it looks like NRCS and maybe some other institutions are going to start doing that in the future. So we have these, directionally understood means, is it improving or is it getting worse? How do I measure that? And then known management practices. What do I have to do to actually improve the thing that I'm trying to measure? And then regional potential ranges are known, which means if I'm farming in Kansas versus farming in North Carolina, how does that change in terms of how I interpret these data? And then the outcome-based thresholds. Do I know the threshold that I need to reach in order to have a positive benefit on my soil? And I'll stop there. So I have a little bit more, but I'll stop there because I wanted to get people to lunch. I don't want you to be angry at me. Thank you for coming to my presentation, and I'll answer any questions that you have.